Hi everybody, today's lecture is about the marine microbial carbon cycle. And I like to do this lecture after I've given my lecture on microbial metabolism, which was the previous lecture, because I need to use those terms and those concepts about different categories of microbial metabolism to talk about what these microbes are doing in ecosystems. And the first one we're going to tackle is the marine carbon cycle. So to use some of those terms like autotrophs and heterotrophs, let's get started. In the ocean, we have these green things that are phytoplankton. Phytoplankton include both bacterial phototrophic organisms as well as eukaryotic. Normally, people call these things algae, but the proper term is phytoplankton and includes both eukaryotes and bacteria. They are photoautotrophs. They're using sunlight, obviously. They're doing photosynthesis to fix carbon dioxide into organic matter. Throughout this lecture, I'm going to use CH2O as an abbreviation for organic matter. More specifically, it's an abbreviation for carbohydrates. You might have learned that this is the ratio of these three elements in carbohydrates, and so it's just a nice shorthand for any biomass, any organic matter made by biology, and in this case, from fixing carbon dioxide. And a byproduct of photosynthesis is oxygen. That's how we got oxygen on the planet to begin with. Of course, this is what we breathe, and we have phytoplankton and trees to thank for the oxygen that we breathe. Okay, now the heterotrophs, specifically the chemoheterotrophs, are microbes that then decompose the organic matter made by the autotrophs. This is exactly the opposite of autotrophy, in that it not only decomposes the organic matter back into carbon dioxide, but it consumes oxygen as it does so. So it's exactly the opposite in every way of autotrophy. So autotrophy and heterotrophy you can think of as two ends of a cycle. And so this is one way to talk about the carbon cycle in terms of carbon fixation by autotrophs, and respiration is the equivalent term for heterotrophs. That is the opposite of photosynthesis. So photosynthesis and respiration are opposites of each other, respiration being the consumption of organic matter, and oxygen going back to carbon dioxide. This is this is a carbon cycle, one version of it, one aspect of it, and in microbiology, we specifically talk about this in the surface ocean as the microbial loop. This term was coined to describe the idea that this can happen very fast. It happens on very short time scales. The cycling of carbon from the atmosphere into the ocean, and then respiration of that carbon back into CO2 and back into the atmosphere again. And so the loop being the carbon going in and out of the ocean and going in and out of multiple members of the microbial ecosystem, phytoplankton and heterotrophic bacteria. And as we continue in this course, we'll meet many other members of the system that are involved in the microbial loop. So the microbial loop, a fundamental concept of microbial ecology, particularly in the ocean, and this is how you should think about it, is the rapid cycling of carbon between carbon dioxide and organic carbon, with photosynthesis and respiration being on opposite ends of that cycle. There are many papers on Canvas about the microbial loop you can read more about, including some classic reviews that go back a few decades now. It's an old idea in biological oceanography. All right, now looking at the carbon cycle from a more global perspective and not only a biological perspective, this is a figure from my introductory biology textbook, and it's highlighting the biological carbon cycles in the ocean and also on land. You see they're roughly equivalent. There's a bit more photosynthesis going on land than in ocean, but you can think of them as roughly 50-50. You can also notice that photosynthesis and respiration on both on land and in the ocean are roughly balanced. Whether there is whether the oceans and the land are a net carbon sink, that is, are they bringing more carbon into the ocean or the land than they are releasing back into the atmosphere? That's actually an ongoing research question. It varies seasonally and perhaps from year to year. And it's a bit of a hard thing to put a definitive answer to. But at the moment, on average, over long time scales, we have been drawing carbon down out of the atmosphere and into the oceans and into stocks of carbon on land. However, at that same time, humans have been pumping carbon back into the atmosphere. And although the number of gigatons of carbon that humans are putting into the atmosphere each year is a relatively small number compared to these numbers. So these numbers are about 100 gigatons, and this is 8 gigatons. That's only 8%. However, it's not balanced, right? We are not bringing an equivalent amount of carbon out of the atmosphere and storing it somewhere as we are pumping into it. So our role in this system is very unbalanced, whereas the natural biological cycles are balanced. And that is exactly the problem. And that is why the amount of carbon dioxide in the atmosphere is steadily increasing since the Industrial Revolution. And this is exactly why, even though the biological cycles are holding steady or maybe even bringing carbon out of the atmosphere. Some terms here, these big arrows are called fluxes. Fluxes are movements of things between reservoirs and environmental sciences. And this is what I just said on annual timescales. Autotrophy and heterotrophy are mostly balanced. That is photosynthesis and respiration. Those were equivalent terms. But the human role is not balanced. So those are fluxes, and this figure is also showing stocks. So that's what I've now circled with my black circles. In the figure, they're in the purple font. And you see these numbers are larger because these numbers are stocks of carbon. These are reservoirs of carbon. These are storages of carbon, whereas these numbers are fluxes. These are movements of carbon per year. So these are per unit of time. These are not a per unit time. That's just what it is, perhaps permanently in the cases of the ocean and rocks. So this is showing different reservoirs, including in the atmosphere. That's the number that's going up steadily since the Industrial Revolution. The amount of carbon in life on land, amount of carbon in life in the ocean, and as well as rocks in the ocean. So for one tangent, let's look at how the biological carbon is divided among organisms. So here's a different figure focusing now just on carbon in biology. And if you look at all of the biological carbon in the world, most of it is stored in plants. And that's because trees are very big, essentially. There are also a lot of grasses. And also when things like trees die, they still have a lot of carbon in them. And it takes a long time to get that carbon back out of even the dead trees. So our world is green, land is green, so that's why this part is most of the carbon is in plants. Second place are bacteria, followed by archaea and fungi. So the microbes are following close behind the plants. Then the animals are a tiny, tiny percentage of all of the carbon in the world. And if you blow up just the animals, then you'll see that most of the animal carbon is in arthropods. So that would be insects mostly. And humans are down here. Wild mammals are here. Wild birds are here. There are a lot of fish, but I also like to point out with this slide that the amount of carbon in livestock is quite a bit larger than all of the carbon in wild mammals and wild birds. So human-raised birds and mammals are taking up a lot more carbon in the world than their wild counterparts. Okay, that was a bit of a tangent. Going back to the marine carbon cycle here, I want to focus now that over time, carbon from land gets washed out into the ocean. The ocean basins collect carbon that runs off from land, and that gets stored into 3 different reservoirs. There's a surface water reservoir, there's a reservoir in marine organisms, which is relatively small because these are all microbial cells. Like the amount of carbon in whales is tiny compared to the amount of carbon in microbes. And then most of the carbon is actually stored in the deep ocean. And that's simply because the deep ocean is really, really big compared to the surface ocean. And over about thousands of years, about 1,000-year time scale, most of the carbon is stored in the deep ocean. So, and then over longer periods of time, that carbon eventually gets stored as rocks. Now we're getting into geology here, and I'm going to skip over how that happens mostly. But the big take-home point here is that on millions of years, on million-year timescales, geological time periods, most of the carbon in the world is stored in rocks. And it takes millions of years for that to happen. Over thousands of years, carbon gets stored in the ocean, and eventually that carbon in the ocean gets stored in rocks. So this is important for understanding because the rates at which carbon gets cycled through biology is really, really fast, but the rates at which carbon gets cycled through the ocean is relatively slow, and the rates at which carbon gets cycled through rocks is really, really, really slow. And if we need to get to the point as a species where we have to take control of the carbon cycle, then we need to understand these different reservoirs, these different fluxes, these different rates. Okay, and I'm going to show you an example of that in this lecture. All right, before I come back to that point about harnessing the carbon cycle ourselves as engineers, let's do a little bit more biology and learn a little bit more about the biological systems and the biological species. The members of the marine carbon cycle can be divided again between the autotrophs and the heterotrophs. And some words we use to talk about the organisms that do autotrophy are phytoplankton, which literally means floating plants. These include diatoms and other things that you can call algae. Algae is not a very good taxonomic term anymore, but it's still a useful term. I still use it. And then the small phytoplankton are cyanobacteria. So these are single-celled bacteria that do photosynthesis. Cyanobacteria are the ancestors of chloroplasts. So diatoms and other eukaryotes that do photosynthesis, they do so because they have chloroplasts, and the chloroplasts came from cyanobacteria. Another way of saying this is that all of the photosynthesis going on in the ocean is done by cyanobacteria or enslaved cyanobacteria as chloroplasts. In fact, that's true of all of the photosynthesis in the entire world because all of the land plants are also using chloroplasts, which evolved from cyanobacteria. So cyanobacteria are really special in that they are the only lineage of life in the history of life on Earth that have ever invented photosynthesis that releases oxygen. So they're really important. There are, of course, other kinds of photosynthesis that do not release oxygen, but cyanobacteria are the only ones that make oxygen while they're doing photosynthesis. Heterotrophy, you can also call the organisms that do heterotrophy in the ocean as zooplankton. This means floating animals. A large zooplankton include things like copepods and krill, and then the small zooplankton are ciliates. As one big example, in other words, single-celled eukaryotes. These are microbial eukaryotes that go around eating other very small cells and eating organic molecules floating around in the ocean. Then heterotrophic bacteria, which are usually considered to be separate from the small zooplankton. That's exactly what they are. They're bacteria, they're heterotrophic. They decompose organic matter or eat organic molecules floating around in the ocean. They are extremely abundant. They, among all of the groups listed here, they are definitely the most abundant in the ocean. All right, some pretty pictures and just doing some fun biology to talk about these organisms. These are diatoms. These are microbial eukaryotes. So these are single cells. They are in domain eukarya. They have chloroplasts to do photosynthesis. And diatoms are special in that they have these glass shells. They have frustules made of silica that are as a protective coating around their cells. There are many different shapes and morphologies, including spheres and square-like filaments and these scary-looking spines. They often occur in chains like this. So they're very pretty organisms. This is a photo of cyanobacteria. This is showing a species of cyanobacteria that often occurs in chains of cells. So it's a single-celled organism, but these single cells are often occurring as parts of large chains of cells. And some of the cells can actually specialize. So here is an example of specialized functional cells along this chain, where some of them are specialized for doing nitrogen fixation. And they have to be specialized cells for that because nitrogen fixation can only occur in anaerobic environments. So this is actually an anaerobic cell where oxygen has been excluded so that it can do nitrogen fixation and provide nitrogen to its sister cells in the chain, whereas the other cells in the chain are doing photosynthesis and releasing oxygen, and you need to sequester that oxygen away from this cell so that it can do nitrogen fixation. So I like to point that out to say that, yes, they're single-celled bacteria, but they still have specialized functions. They have cellular differentiation. You might even want to call this a multicellular organism in the way that it is living its life cycle. And we'll come back to that concept later. But for now, those are cyanobacteria. They do a lot of photosynthesis in the ocean. These are small zooplankton. These are things like copepods and krill. These are ciliates. So these are single eukaryotic cells. They're called ciliates because they have all these cilia that they use to create ocean currents that bring particles into their mouth-like structure on the side of their cell. So it's a single cell, but it has something that looks like a mouth, which I think is pretty cool. This is also a ciliate. They're really important grazers of bacteria in the surface ocean. All right, so we've got our diatoms and cyanobacteria and other phytoplankton here in the surface ocean. They're doing autotrophy. They're fixing carbon dioxide into organic matter. We already talked about how approximately half of the carbon fixation occurs in the ocean and half occurs on land. We've got our heterotrophic bacteria that are completing the microbial loop with photosynthesis and respiration occurring and balancing each other in very short timescales, and timescales of days even, and at most years. Okay, so this is a loop. I've told you a few times now, this is a rapid loop. It cycles everything back and forth really quickly. If that's the case, then how did oxygen ever accumulate in the atmosphere in the first place? For half of Earth history, there was no oxygen on the world. It was entirely anaerobic. That was the heyday of anaerobic microbial metabolism. It was the time when most of the metabolic pathways on Earth evolved in the absence of oxygen. And then at some point, about halfway through Earth history, 2.3-ish billion years ago, oxygen suddenly appeared. It at first was almost certainly a poison that killed off almost everything. Perhaps the greatest mass extinction event ever, although it's very hard to get good data on that because those microbial cells do not leave very many fossils. But after that, the world was different, and now aerobic metabolism was the name of the game. If you could breathe oxygen, you had a huge metabolic, energetic advantage over your competitors. But how did that happen? How do you suddenly get a big accumulation of oxygen if the microbial loop is cycling that oxygen as soon as it is produced? Well, what you have to do is sequester the organic matter from the oxygen. So the microbial loop operates by burning the organic matter with oxygen. And if you're going to allow that oxygen to accumulate and not be immediately consumed, then that means you also have to get rid of the organic matter so that it can't react with the oxygen immediately. Today, that happens basically via fish poop or other zooplankton poop, or more technically speaking, zooplankton fecal pellets. It turns out the zooplankton fecal pellets in particular are designed to be hydrodynamic. That is, they very efficiently sink to the ocean floor, and that operates as a very effective biological carbon pump. It is pumping carbon from the surface of the ocean to the seafloor via the sinking fecal pellets. And that is how today we have a net surplus of oxygen in the atmosphere. That's why we can breathe oxygen. But we know that oxygen started accumulating in the atmosphere about 2.3 or more billion years ago, and there were definitely not any animals making fecal pellets back then. So how did it work back then? Probably through thick algal mats. So some kind of thick mats of phytoplankton that were probably covering the surface ocean in these big green mats. And perhaps because they were just so heavy, these big green mats were able to sink to the ocean floor faster than they could be consumed by oxygen. It's a bit of speculation. There's some geological evidence for it. But it's basically the only way that anyone can imagine that you could sequester this amount of organic matter away from the oxygen to allow the oxygen to accumulate. So that does not solve the problem. This is an ongoing major research question. What did the ecosystem look like where oxygen first started accumulating in large quantities? We really don't know exactly the answer to that question, but it must have involved some kind of carbon burial. That's the key, is you got to bury this carbon so that it does not immediately get burnt up by the oxygen, allowing the oxygen to accumulate. Now, if you have a biological carbon pump operating to bury the carbon on the seafloor over thousands or especially millions of years, and that carbon stays in the marine sediments for millions of years and basically turns into rocks, then basically what you're doing is you're making oil. This is petroleum. Petroleum is buried organic matter that used to be on the surface of the ocean. All of the oil that we're burning right now used to be phytoplankton and heterotrophic bacteria in the surface of the ocean millions of years ago. So that's kind of the overview of the carbon cycle from this kind of perspective of the microbial players of the carbon cycle. So I haven't really told you much new that I didn't already mention in that big overview of the global carbon cycle. I've just now told it to you from the perspective of the biological species that are doing the work, with a bit of perspective from a billion-year time perspective of how oxygen accumulates in the atmosphere. Okay, so let's complicate the story a little bit more by bringing in some more species, because we're biologists and we like to do that. Let's talk about coccolithophores. Coccolithophores are eukaryotic photoautotrophs. They are members of the phytoplankton. They do photosynthesis. They have chloroplasts. But they're also white because they have these calcium carbonate shells. They make these shells around them. So don't confuse these with the diatoms. The diatoms have glass shells. These are carbonate shells. In other words, chalk. When the coccolithophores die, their carbonate shells sink to the bottom and you form massive carbonate deposits on the seafloor. You can see the carbonate deposits as the White Cliffs of Dover. I've mentioned this before. Here's a big coccolithophore bloom as seen from space, probably being infected by viruses. And when the viruses infect the coccolithophores, they release the carbonate shells. The carbonate shells sink to the bottom, and over geological periods, you can form massive carbonate deposits like you see in southern England. Okay, now let's imagine what happens to these coccolithophores when the ocean becomes more acidic, which is what is happening right now, because we are interacting with the carbon cycle in an unbalanced way, and we are pumping carbon into the atmosphere much faster than we take any carbon out of the atmosphere. This accumulation of CO2 caused by humans is making the ocean more acidic, because when carbon dioxide dissolves in water, it becomes carbonic acid. So additional CO2 inevitably means more acid in the ocean, just with simple chemistry, because dissolved carbon dioxide is an acid. This drops the pH of the ocean, and that's really bad for lots of reasons, and specifically to this story, because the coccolithophores depend on a very delicate pH balance to make their carbonate shells. And if they can't make their carbonate shells, then they get very sick. Here are some images of sick coccolithophores who are making malformed carbonate shells because they're living under very poor pH conditions. And this is not theoretical. Although these images are from an experiment where they artificially lowered the pH to see what would happen. This is not theoretical. You can actually measure the acidifying ocean right now. It's happening right now. These are a few different locations in the ocean until 2015, so it's already much worse than this. This is already out of date by several years. But you can see, for example, looking at Hawaii, as CO2 in the atmosphere goes up, the pH of the ocean goes down. And these units may not look too impressive to you. So since 1990, around Hawaii, it went from 8.12-ish down to 8.05-ish. But remember, the pH scale is logarithmic, so every unit is tenfold times more acidic. And organisms like coccolithophores are really adapted to very stable pH, because the ocean is a really, really, really good pH buffer. That is, it's really hard to make the pH of the ocean change very much because it is so well buffered. And a lot of living things like coccolithophores are adapted to that very strong buffer. And if we make it so unbalanced that the the pH buffer of the ocean is fundamentally changed, then that's going to have massive biological consequences, and these coccolithophores are one example of that. The ongoing research on these coccolithophores actually makes it a little bit more complicated. We may discuss a paper later in the semester that shows that some species of coccolithophores may actually be adapting to the lower pH, but that's a question mark. That's like an interesting research question. Overall, it's pretty clear that it's a very challenging set of conditions for coccolithophores to adapt to. Okay, so that was the one very quick version of the scary nightmare story. So let's talk about what can we do about it. I get this question a lot. Can we manipulate the carbon cycle in some way now that we seem to understand it so well to make things better? And this is not a new thought. People have been thinking about this for a while, and they've even tried to do it. So that's the story I want to tell now, is a story of actually trying to use this biological carbon pump for our own purposes. So the places where you might want to try to do something like this are these places called high nutrient, low chlorophyll regions. They are places in the ocean which are huge, where you have a lot of nutrients. That is, you have a lot of things like nitrogen and phosphorus, basically ocean fertilizer, where it should be possible for phytoplankton to grow in high densities and do a lot of photosynthesis and bring carbon out of the atmosphere. However, these are low chlorophyll regions, as shown in this map. Blue means low chlorophyll, red means high chlorophyll. And in these big open spots in the ocean, you have very low chlorophyll, meaning very low amounts of phytoplankton. So not much photosynthesis is going on in these low chlorophyll regions, even though we know that there's actually a lot of nutrients out there. So what's going on? Why are the phytoplankton not growing? And it turns out it's because there are other things that they need besides nitrogen and phosphorus, for example, iron. So it turns out that the big interiors of the ocean are often limited by iron, and that's because iron comes from land. Especially a big source of iron to the ocean is being blown from deserts. So if you are downwind of a desert, you have a lot of iron. If you're very far from any deserts, you're almost always iron limited in the ocean. So the idea is, why don't we just dump a bunch of iron into the ocean and cause a phytoplankton bloom, and that will cause a bunch more carbon dioxide to be fixed and brought out of the atmosphere, and maybe by just dumping some rusty nails into the ocean, we can solve our climate change crisis. So this experiment has been done multiple times, in fact, and that's basically what they do. They load up a ship with a bunch of iron shavings to maximize surface area, and dump these rust buckets into the water, and then do some ecological studies to see if that results in a big phytoplankton bloom and uptake of carbon. And now that you understand a little bit about the microbial loop, what do you think will happen in this experiment? So one prediction you might make is that yes, you will have a bloom of phytoplankton, you will have more photosynthesis, you will have more carbon fixation, and then, because you know about the microbial loop, what will happen after that? Well, it's not only photosynthesis going on, right? You also have respiration. So if you have a big generation of organic matter by photosynthesis, and you still live in an oxygenated world, then you're going to have a lot of respiration as well. And this is indeed what happens, is that when you do these experiments, you stimulate the phytoplankton, but you don't only stimulate the phytoplankton, you stimulate the whole ecosystem. So you supercharge the whole microbial loop, and very quickly you bring carbon dioxide out of the atmosphere into the ocean, and just as quickly you pump that carbon right back out into the atmosphere. And you are back to your steady state, because microbial ecosystems in general are balanced, and if you just make them run faster, then they run faster, but they're still balanced. So it's really hard to make anything go in one direction when you're talking about natural microbial ecosystems. So that's the microbial loop. And that was basically the result of these experiments. So how might we do this experiment different? Or is there something else we can do to actually store this carbon on the seafloor? How can we actually bury this carbon? Just adding iron, just stimulating the phytoplankton is not necessarily going to result in burial of the carbon. The idea with these experiments is that if you could make a thick enough mat of phytoplankton that it would just sink to the seafloor on its own, it turns out that just doesn't really work, or it's not practical. In part because we also have things like fish nowadays, unlike 2.5 billion years ago, that will eat those mats of phytoplankton. So it doesn't work to just do it in the simplistic way. But what if we could? What if we did find some way to pump a bunch of organic carbon as a result of carbon fixation to the seafloor, so we actually sequestered it away from the microbial loop and it did not immediately go back into CO2? Well, how long are you going to have to do that, right? Is the question. Basically, in order to really make this work on any time scale that would actually be useful, basically you're going to have to bury it in the same conditions that are also required for forming oil, right? This is basically what you're really trying to do if you're going to go down this road of trying to harness the biological carbon pump for your own purposes. Basically, what you're saying to yourself is, what I really need to do is to store this carbon so that it eventually becomes oil, which I think obviously begs the question of why don't we just stop digging the oil out of the ocean in the first place, and then the problem is solved. Obviously, that's a hard thing to do, but it is interesting to note that in order to solve this problem that we have created by digging oil out of the ocean, this idea is basically, well, why don't we form more oil? That raises a whole bunch of social and political and engineering issues, but let's go back to the biology for a minute. Let's say that we do this. We store a bunch of carbon in the seafloor. And let's think about the short-term biological consequences of that, even if we decide it's something that we want to do. So if you have more carbon on the seafloor, then there are also bacteria on the seafloor, right? We've been focusing on the microbial loop, which is the surface ocean, but you've got bacteria everywhere, especially heterotrophic bacteria. They're everywhere in the world. They are always going to consume organic matter wherever it is. So you're still going to make CO2 in the deep ocean. And if you've got a whole bunch of organic matter here, remember the whole idea is that you've got more organic matter being formed than you have oxygen to burn it. All right. The idea is you have a surplus of carbon. So if you have more carbon than you have oxygen, then all the oxygen goes away. And we learned in the previous lecture that when all the oxygen goes away, then what happens next? Well, you use the next available electron acceptor, which might be nitrate. The nitrate goes away really quickly as well. Then you start using things like iron or sulfate or CO2. So if you succeed in burying a bunch of organic matter on the seafloor, some of it will be consumed by the aerobes and the denitrifiers, which are the bacteria that eat nitrate. And then once that is gone, what you are now doing is you're making things like hydrogen sulfide and methane with sulfate-reducing bacteria and methanogens. So you've now created a dead zone on the seafloor. There's no oxygen because you've consumed all the oxygen with this big lump of organic matter that you've deposited on the seafloor. Not only is there no oxygen, you're now making things like methane and hydrogen sulfide. And so now you've got a really nice, yummy anaerobic microbial metabolism, which is great for anaerobic microbiologists like me. There's lots of things for us to study, but there are no more fish because fish can't survive in conditions like this. So that's probably not a good outcome for most things that we care about. Another physical consequence of this is that, of course, the ocean is getting warmer at the same time. And a consequence of physics and chemistry is that warm water can hold less gas than cold water. So as the ocean gets warmer, it can hold less oxygen, just due to that simple principle of chemistry. And so the problem gets compounded. So not only are you consuming oxygen if you're burying organic matter, but the ocean is already holding less oxygen. And that's a dynamic that you can already see, and you can already read papers about how the decreased capacity of the oxygen to hold oxygen is leading to massive fish die-offs, for example. Okay, so to kind of summarize this, in principle, this is not a completely stupid idea to somehow stimulate the biological carbon pump. You just have to remember that there is this thing, the microbial loop, which will cycle things really quickly. And if you somehow solve that problem and you bury a bunch of carbon on the seafloor, you've got to preserve it on the seafloor in some way, which basically means that you got to make artificial oil. And there are just so many factors in play here that whenever we are thinking about trying to control the entire ocean for our own human engineering purposes, then I think it's maybe time to think more deeply about what exactly we're trying to accomplish. So that's my lecture on the biological carbon cycle and the microbial loop and a little bit of current events. And we're going to continue this story in more fun biological and ecological ways in the next few lectures. So I'll see you then. Bye-bye.